Translation is the process of polypeptide or protein synthesis. Polypeptide is more correct because you are actually building peptide bonds between the different amino acids, and while all polypeptides are proteins, not all proteins are just one polypeptide. It has 3 different stages: initiation, elongation, and translation. I'm going to go over all 3, and I'm also going to post some short videos showing you how the ribosome works and how antibiotics interact with it. Step 1 is initiation, where to start. And that really is the big problem. Where should translation start on the messenger RNA? If you remember, we talked about the messenger RNA has a 5' untranslated region, so it's not coding for a polypeptide, and a 3' untranslated region at the other end, so it's not starting at the first 3 letters of the messenger RNA, it's starting a little ways in. So the real issue is, where should that first tRNA carrying that first amino acid go along the messenger RNA, and how will it actually get there? How will it find the right one? And I'm just going to remind you that the part of the protein that codes for the polypeptide is called the open reading frame. And the first codon that translation starts at is called the start codon, and for most organisms this is going to be methionine. It is specified by AUG, and there's actually a special different tRNA that inserts that very, very first methionine. It's a modified tRNA. Note that Shine-Dalgarno sequence in bacteria because we're going to be coming back to that. Prokaryotes and eukaryotes use slightly different mechanisms to get the ribosome over that first start codon and put it in the right place on the messenger RNA. In prokaryotes, that is really what this Shine-Dalgarno sequence is for. It is going to directly interact with the ribosomal RNA in the small subunit of the ribosome, binds by complementary base pairing. Again, it's anti-parallel, and that binding helps position that small subunit of the ribosome in the right place to start translation at that start codon AUG. Once that pairing takes place, that 30S ribosomal subunit and their associated initiation factors and that initiator tRNA will be positioned and form over that start codon. In eukaryotes, it's a little bit more complicated. It requires the 5' cap, and also the 3' poly A tail plays a role. Initiation complex is going to form at that 5' cap. It contains the small subunit of the ribosome, which is 40S in eukaryotes. It contains that special initiator tRNA that is going to carry methionine. And it also contains a whole host of initiation factors that are not shown here. There's at least 11, so it's a lot more complicated. The 40S ribosomal subunit and initiation factors, they're all going to form at that 5' cap, and then they're going to actually move along and scan the messenger RNA until they find that start codon, that AUG. Once that happens, the entire ribosome assembles. And I should have said, in both prokaryotes and eukaryotes, once you are surrounding that first start codon and that initiator tRNA carrying methionine is locked into place with the small subunit, the large subunit is going to pop on. So either way, you're triggering ribosome assembly, and those initiation factors dissociate and move off to start new ribosomes. The next step in the process is elongation. And remember that when this whole process starts, that first tRNA, the special modified one carrying methionine, is already sitting over the start codon, and they're occupying the center of the ribosome, what is called the P site. So during elongation, the first normal tRNA is going to enter through the A site, and that is where all of the tRNAs actually enter the ribosome. And I've circled it here so you can see exactly what I'm talking about. There's a little intake funnel, and basically what happens is lots of different tRNAs get tried on for size to find the one, find and reject, find and reject, find and reject, find the one that is actually going to complementary base pair to that triplet, that codon in messenger RNA. Once that codon finds a match, that tRNA finds its partner in the messenger RNA, the amino acid it carries will be added to the chain that is currently sitting in the P site. And when that starts, this chain will only have that single methionine. But as it keeps going, there'll be more and more and more amino acids attached to the tRNA that currently occupies the P site. It is joined, that new amino acid, into the chain of amino acids by the formation of peptide bonds. And this is just showing you where that P site is, that central site. The third position in the ribosome is called the E site. And that is where the tRNAs that are finished leave the ribosome. And you'll notice they're called deacylated tRNAs. That means they no longer carry an amino acid, but they can actually be recharged with the new correct tRNA using those amino acyl transferase enzymes we talked about last time, and recycled to reparticipate in translation. So I'm just going to go over how that process actually goes from the beginning, and I'm also going to show you a short movie of this. As we said, the process is going to start off with a tRNA, that special initiated tRNA, sitting in the P site carrying methionine. The next tRNA is going to bind to the A site and enter. Notice that green tRNA that is carrying an amino acid currently occupying the A site. That little thing called EFTU, that is an elongation factor. So again, there are many accessory proteins that help in this process. I'm not showing them for simplification here, but some of the videos you see will include them. Once it finds a match, a peptide bond is formed, and what happens is the ribosome actually, it's called translocation, it's going to move over. Everything in the ribosome is going to move over one spot. So what is in the P site will move to the A site, and it will no longer be attached to its amino acid, so it is free to be recycled, it is uncharged. What was in the A site will move over to the P site, carrying the peptide chain as it's growing, and the A site will now be empty. Once it's empty, you can now add a new tRNA. And again, it's just going to be many tRNAs are going to come into that site. It's a blind process, so each of these tRNAs is just basically going to be tried on for size until you find one that is able to complementary base pair and stabilize that tRNA in the A site. And then the whole process will begin again. Its amino acid will bind by peptide bonds to the amino acids that are in the P site. Everything will move over one spot. The one that was in the E site will exit. The A site will now be empty, and a new tRNA can enter, and you can carry on. And that is elongation. The last step in the process is termination. So how does it know where to stop? And remember that 3' region is untranslated. So it's not just that it's going to run to the end of the messenger RNA. Again, there's an actual code that was originally in DNA, got transcribed into RNA, that is going to tell the ribosome where to stop. And that code is called a stop codon. So termination has 3 kind of main parts to it. First of all, that stop codon has to be recognized by a release factor. And these release factors are small proteins. They are not tRNAs. And that release factor, there is no tRNA that corresponds to the stop codon. It is going to bind to that stop codon. When that happens, it causes the polypeptide to be released. So at that point, it is done. You have finished the polypeptide or protein that the ribosome is making, and the other tRNAs are released. Again, they can be recycled to be used to make more proteins. At that point, the ribosome is going to disassemble. So the small and large subunits separate. They'll be brought together again when the next ribosome assembles, and the messenger RNA is released. That might be used again to create a brand new protein, or it might be broken down. There are plenty of enzymes, as we talked about, that degrade the messenger RNA. And that's one way of regulating gene expression, actually, how long a messenger RNA persists in a cell. The last thing I wanted to say is that most messenger RNA molecules can be translated many times. And these images just show you in the middle, the kind of line going along the horizontal line is the messenger RNA. The large kind of round circular balls that are attached to it, these are ribosomes. And the point is that multiple ribosomes are attached to that messenger RNA, and they're moving along it from 5' to 3' at the same time. So while the one at the end, when it gets to the 3' end and releases the messenger RNA and disassembles, the other ones are still going along the process and attached. And these kind of areas, these factories where multiple ribosomes are working on the same messenger RNA, are called polyribosomes or polysomes. And that is it.